Okay, I won't be offended if anyone wants to go and play cribbage either. So, yeah, so tonight I'm going to talk about the gut microbiota, and in particular how the gut microbiota can interact with our brain and how it does this and what are the consequences of this. I wasn't entirely sure of the audience, so I've kind of started low with the idea to build up, and I'll kind of, based on how many people are asleep, I'll try to adapt as I go. So, starting from the very beginning, we're talking about microorganisms. And obviously, the study of microorganisms is microbiology. And the main microorganisms we're going to study or talk about tonight, well, we cover the bacteria, really. Most of my talk is going to be on bacteria. But when we talk about the microorganisms, we're also covering the archaea, which are very similar to the bacteria. The fungi, mainly the yeasts, when we're talking about the microbiota. The viruses. And we also cover the protozoa as well. And actually, we usually throw the worms in there as well when we talk about the microbiota. So I'm trying to get out of everyone's way. Probably, where's the best place to stand here? Okay, so when we're talking about microorganisms, we're talking on the micrometer scale, which is 1,000th of a centimeter. So to put this in perspective, you think a human hair is around 80 micrometers wide, and a bacteria is about 2 micrometers long. So it's about 40 times smaller than the width of a human hair. Viruses are actually much smaller. So just kind of like setting the scene there. So important point to note then, our human body is colonized in and out by a vast array of these microorganisms. We refer to these as the microbiota. And different places have different composition, different types of microbes. So we have an oral microbiome, we have a lung microbiome, we have a skin microbiome. There's lots of companies trying to develop skin products to look at the skin microbiome. We have the gut microbiome, which is the majority of things I'll talk about tonight. There's even things like the vaginal microbiome. And all these different niches have different communities of these microorganisms. Again, covering viruses, bacteria, and different fungi. And in this context, you, the human, are the host, and then you support these microorganisms. I think my phone is off. So the human is the host, and the microorganisms, you're supporting them, and they're usually given some kind of benefit in return. A really important point to hammer home is that it is unique to you. So everyone in this room has their own unique microbiome, and I'll go into why that is. But that's really important to remember, because as I go through some of the benefits, everyone has a different microbiome, which might mean different benefits, different outcomes. So just in terms of terminology, you probably see these two terms, and to be honest, most people use them interchangeably. There is a subtle difference. So if we talk about microbiota, we're generally referring to the complete composition of who's there, whereas microbiome is a biome. It tends to refer to the complete, so it's who's there, also their genomes, also the environmental conditions. But to be honest, they're pretty much used interchangeably. So just some facts then. The human body, and these get revised up and down all the time, but the current state is that there's around 39 trillion microbial cells in and on the body. And so that means that they outnumber the human cells by about 1.3 to 1. So there's more microbial cells in and on your body than there are of your own cells. So you're more microbial than you are human. And they tend to exist in a symbiotic relationship. So you give them somewhere to live, like you keep them safe, you also give them food, and in return they provide you with some benefits. You can also live in a commensal relationship where it's a bit more, they just exist, but they cause you no harm. The current estimate is, again, it's about 3 pints, so your microbiome is about 3 pints if you put them all together. And the majority reside in the gut, and particularly in the large intestine, or what is sometimes called the colon. So that's why you might hear of it called the colonic microbiota. So I said you're all unique, you've all got this unique microbiome, and that's because of all these pressures, or the pressures on this community. And so you acquire, there's a lot of debate actually in the field about when you actually acquire your microbiome. Some people thought you can't be sterile at birth, you must have it from your mother. But then they found out what people were measuring in newborns or in certain fluids was actually just contamination in the kits. So currently people think that you are sterile before you're born. You might get some exposure to fragments, but you're pretty much sterile. So as soon as you're born, you enter the environment, you start to be colonized. Then you have an infant microbiome, which changes by about the second year of life, as you then move to a child, to an adolescent microbiome, and then to an adult microbiome, and then as you move to later life, you have a more aging microbiome. So things like changes in dietary preferences, physical activity, immunosenescence, like decline of immune system, all kind of impacts on a declining microbiome. But so at birth, one of the main factors is your host genetics. So your genomes, your genetics, cause you to express these glycans, which are like sugars, throughout your gut. And they serve as kind of signposts. These are like sugars that the initial colonizers will metabolize. And so that sort of serves as these signposts for them to start growing, and then you get communities forming on top of those. Mode of delivery is really important in early life. So the vaginal microbiome is dominated by lactobacilli. So vaginal born babies, their first microbiome is dominated by lactobacilli. Whereas C-section born babies, their microbiome is more reflective of the skin. So they tend to have a much more diverse microbiome. Then we have breastfeeding. So mother's breast milk contains these particular sugars, so human milk oligosaccharides, and these can't be broken down by the baby. So what happens is they make it down to the colon and they promote the growth of particular bacteria, particularly the bifidobacteria. And so a lot of these changes disappear once the baby's weaned, once they get a bit older. And then factors like diet, so whether you're a meat eater, vegan, omnivore, vegetarian, this can impact. Obviously, antibiotic intake will directly impact on your microbiome. Infections, also environment. So we know that if you've got a dog, you tend to have a different microbiome to people without a dog. Environmental pollutants can also change your microbiome as well. And then there's things like psychosocial stress. So if you're really stressed, that can impact on your gut environment, which can impact on your microbes. So all these pressures are adding up to mean that everyone is a slightly different composition. And so this tends to be what a healthy gut might look like. So this is in your colon. And so you have these host cells. This is your gut wall. And these are all nicely, tightly packed together. And they're joined by things that we call tight junction proteins. And so what that means is they cause this really tight barrier. So stuff can't move between the cells. They have to go through the cells. And the reason why this is important is it means your cells actually have a lot of control over what passes from the gut into the blood. On top of that, these release mucus. So you have this tight mucus layer, and that serves as this physical barrier to stop bacteria or microorganisms moving across. And then on top of that, you have a more diffuse mucus layer. And it's here that a lot of microbes will live. And so if you've got a healthy gut microbiota, you're probably going to have a high amount of fiber in your diet. So if you take one thing away from this, eat lots and lots of fiber. So fiber's good. And the reason why fiber, particularly non-digestible dietary fiber, is because you sort of lack the enzymes, the ability to break that down. So that means as that passes through the gut, one, it leads to fecal bulking, so you have a lot more material passing through your gut, reduces transit time, so the amount of time stuff takes to go from your mouth out into your poo, and that can be good for your health. But also this fiber, because you can't break it down, it becomes food for these microorganisms. And the carbohydrate or fiber degrading bacteria tend to be the good guys. They break this down into chemicals which can be good for health. And so this is just one particular one. Don't worry too much about this for now. But they can break down these fibers. They can increase their mass of these good bacteria, produce this chemical. And these chemicals, these good chemicals, can do things like they can improve these tight junctions, this integrity of the gut. They can acidify the gut, which makes it more protective from pathogens. And they also have properties like they can reduce inflammation. They can have anti-cancer properties as well. So that tends to be what a good gut looks like. A more sort of dysbiotic or harmful gut, if you have low fiber in your diet, or if you have a sort of a slightly negative composition of bacteria, then what can happen is they can break through, they can degrade this mucus layer, move in that physical barrier, so there's more direct contact between the microbes and the gut wall, and that can trigger things like inflammation. They can also release toxins which break down these tight junction proteins, And so all of a sudden you lose that ability to block out things. And this is what we often refer to as leaky. Is it because I'm standing too near that? This can lead to something called a leaky gut, which is something we see as people get older. And so you lose that ability. And one of the consequences of that is it can cause inflammation, which again can be adverse for health. So that's kind of like in the local gut, just the gut health. A really important thing with the microbiome is actually its metabolic capabilities. So as I say, it's about 39 trillion cells in and on the body. And if we just really consider the bacteria that are in the gut, a really important point to note is that each individual bacterial cell brings with it an accompanying genome. Okay, and there are trillions of genomes then in your gut. Different bacterial cells bring with them different genomes. So if you think about all these trillions of different bacteria, you start to see there's trillions of different genomes. Each of these contain different genes. So if you think about all of these genes collectively, we refer to this as the metagenome. I'm throwing lots of terminology at you now, I know. But metagenome, basically the mass of all these genes. It's been estimated that there are 3 million different microbial genes in the metagenome. So the human genome is around 24,000 genes. So anyone that's good at maths can work out that that is about 100 to 150 times more genetic information or genes in the metagenome than the human genome. That's a huge amount of extra information. And a lot of these genes will encode enzymes which will perform metabolic functions. So what the microbiota do is they provide this sort of extension, biochemical extension to our limited genome. And a really important point to mention here is that the microorganisms were here first and we evolved around them. So we have co-evolved in the existence of these, and so really we're so dependent on the microorganisms and their metabolic activity to function. So what happens is this mass of different metabolic functions allow us to process a lot more dietary input, different diets, and it exposes us to a lot more metabolites. And so this metabolic activity means, as I've just said, helps to expand our flexibility and be able to digest different inputs. It allows us to extract calories from the diet that we otherwise wouldn't be able to get. So if we didn't have microorganisms, that dietary fiber would just pass straight through our bodies, but because the microbes are there, they can extract that energy. They can extract more calories. They also have an impact on drug metabolism. So if all the people in this room, you've got a microbiota that breaks down, let's say, paracetamol, and this side doesn't, then if we give you all paracetamol, it won't have an effect on you because your microbes will break it down before your body can absorb it, whereas we will see an effect of the paracetamol. So we think that the microbiota contribute to variation in drug responses. Already talked about gut health, metabolic regulation, also pathogen exclusion. So most pathogens are not very good competitors in our gut. So normally if a pathogen comes along, it won't be able to establish in our gut. And that's because the microbes that are already there can outcompete it so well. One of the things you might have heard of, C. difficile or Clostridioides difficile. So a sort of a healthy gut is very robust against fighting C. diff off. It can't establish in a normal gut. If, as you get older, your gut declines, your gut microbiota declines, that's when C. diff can establish. Or if you take get given a lot of antibiotics, that suppresses your microbes. Your C. diff can get in, and once it gets in, it can release a toxin that suppresses the other microorganism. It's very hard to get rid of. So the pathogen exclusion or barrier function of the microbes is really important. We also know it's really important for training the immune system as well, and there's links between the microbes and things like allergies. So because of this really central role in metabolism, this is why we often think of the human body or us as supraorganisms, because we are the sum of all of these interacting genomes, sometimes referred to as holobionts as well, which is kind of like this sum of all these genomes together. So we're a superorganism, and these interactions between your genome, your microbiome, they're changing throughout life. and they have a big impact on health and disease. And we even think it's not just when you're born. There's also evidence now that actually your mum's microbiome is having a big impact on future development as well. And we're even studying how this can happen at preconception. And so there's studies underway now giving things like pre and probiotics to see how this could, to the mum before she's pregnant, to see how this can impact on development. Now, from a metabolic point of view, the majority of substrate or food that the microbes get comes from our dietary intake. It's that or our secretions into our gut. So different microorganisms can break down all these different dietary inputs. And what happens is they produce different compounds, different chemicals. And this can have a couple of different effects on the host. One is substrate bioavailability. So if your microbiome in general is breaking down more amino acids, say this side of the room, you're breaking down more amino acids than you. Everyone eats a big high meat diet. All those amino acids get broken down here. You don't see those amino acids, whereas you receive all those amino acids. So nutrient flow, the microbiota can influence what the body sees from the diet. Also, we have something like called receptors, and again, this is a really good illustration of how we've co-evolved in the presence of these microorganisms. If you think like a lock and key, so your body has these locks expressed throughout the body, and when the key or the microbial metabolite, so these locks are specific to microbial metabolites, so when the microbial metabolite binds to the receptor in the host, it causes a set of genes to be expressed which changes different metabolic activity. And so the microbes can also impact on the human body by producing these signals that bind to receptors expressed by the body. So they can change host metabolism locally in the gut, but these metabolites can also enter the blood and go throughout the body where they can bind to these receptors. And so these can impact a whole range of pathways, including on the brain. And so this brings me on then to the gut-brain axis, which maybe some people have heard about. This is kind of growing in the news all the time. So there's a long time we've known that the brain can influence the gut. So a prime example of this is when you're nervous and you get things like butterflies in your stomach. That's your brain, the stress of your brain changing the environment of your gut, which will have an impact on the microbes. It can actually change your microbes. There's now evidence that the microbes can also influence the brain, though. And there's been examples of this in nature known for a long time. So this is one example. These are what we call zombie ants. So this is a particular type of fungus. I think these are in a rainforest in Peru. So this fungus wants to live in a particular part of a tree because that's optimal growth conditions. But it can't get up there. So what it does is it infects an ant, takes over the central nervous system of an ant, causes it to climb up to a particular good spot on a tree. The ant bites down on a leaf or bark or part of the tree, and then it dies, and the fungus can grow out the back of the head. So that's a good one. That's actually been made. There's a movie called Last of Us. I think that's based on these. This is another example. This is Toxoplasma gondii. This is a single-cell pathogen. Causes toxoplasmosis, one of the most widespread, most prevalent infections in the world. And so toxo can live in rodents, and to complete its life cycle for sexual reproduction, it needs to get into the cat. The problem is mice are scared of cats, so what it does is it changes the central nervous system, and instead of being sort of scared of the odor of a cat, it actually causes the mouse to be attracted to the odor of cats. So it seeks out the cat eats the mouse, and then the toxo can jump into the cat. Another example is this particular hairworm. It gets into these grasshoppers, but then the worm needs to get into water again to complete its life cycle, so it causes the grasshopper to seek out the water and then effectively commit suicide, dive into the water, and then the worm escapes. So lots of examples in nature of how microorganisms might influence host brain. And so this started probably about 20 years ago now, a set of experiments to look at could the microbiota change behavior. And one of the ways to study this is by looking at germ-free animals. Now, how you look at a germ-free animal is you take usually rodents, sometimes mini pigs, Need a bit of a bigger cage though. But usually rodents. So you would breed mice or rats in sterile conditions, so they don't receive any exposure to any microorganisms, and then you raise them. You create this breeding colony. They're really hard to maintain, but what this means is you have a germ-free rodent which doesn't have any exposure to the microbiota. It doesn't have a microbiota, and you can compare this to normal, sometimes called conventional animals. Here they're called specific pathogen free. It's just like another term, so we just know they don't have pathogens, but these have a microbiota, these don't. And one way to test memory is this novel object test. So in a cage, you start with a training day or a training setup where you have two objects in the cage. And mice are quite inquisitive, so they'll spend a lot of time like sniffing, playing with these two objects. And then on the testing day, you'll take one away and you'll swap it with a new or a novel object. And the idea is, if the mouse has memory, it will say, oh, well, I actually played with this one yesterday. This one's a new one, or I'll spend a lot more time looking at that. So we can see when you've got a microbiome, the mice spend about 70% of their time playing with the new object, showing they've remembered that the other one they played with yesterday. But in the germ-free animals, you can see they spend 50% of their time with either object, showing they lack that memory. So without a microbiome, there's an impairment of memory. Another way to test this is this T-maze spontaneous alternation test. So it's kind of like a maze. You start the mouse here. Usually or sometimes there's a treat on one of the sides. So you start the mouse there. You look and it will go left or it will go right. If it remembers, it will change direction on the next go. So they will alternate. So again, if you've got a microbiome, you alternate 80% of the time. If you don't, if you're a germ-free without a microbiota, then they alternate only about 40% of the time. Again, showing that they lack this memory. Another way to test emotional behavior, this is the open field test. So this is like a big white box, and you put the mouse in there. And the idea is if they're sort of low anxiety, they'll spend a lot of time running around, probably spend more time in the middle. Whereas if they're more anxious, they'll spend more time at the sides. So you can see here we've got germ-free on the bottom with a microbiome on the top. You can see the germ-free animals spend a lot more time moving around and also more time in the middle. So they actually, if you don't have a microbiota, you have lower, anxiety-like behavior in these mice. Another way to test this is what we call the elevated plus maze. So it's like a giant plus sign raised off the floor, where two of the arms are what we call closed. They have sides on them, and two of the arms are open. And the idea is if the mouse is more anxious, it will spend more time in the closed arms. If it's less anxious, it will spend more time in the open arms. And in particular, they'll spend more time at the very ends of the open arms. So you see here, germ-free in black, they spend significantly more time in the open arms and significantly more time at the very ends. So again, the germ-free, if you don't have a microbiota, significantly lower anxiety-like behavior. Really interesting point with this study was that they found that so you can take these germ-free animals and put them in a cage, either in a normal lab environment, or sometimes mice and rats are coprophagic, so they'll eat each other's poo. And so what that means is you can actually swap microbes between animals. What they found is if you colonize the animals before they're 6 weeks old, you can reverse the behavior. But if you only colonize them after 6 weeks, that behavior is sort of set in stone. So there's a critical window where this microbiota can influence this brain development, this behavior. And we're really interested in this. So the majority of brain development happens in the first 2 years of life. And that's also a critical period of when you're acquiring your microbiome. We talked earlier about how things like birth delivery, early life nutrition, all impacts on how you colonize. So there's a lot of research going on now trying to see how different impacts influence the microbiota development, how that translates into brain development. So in terms of humans then, there's a lot of, and this is what a lot of the initial motivation for studying this connection was, there's a lot of things like comorbidity. So for example, people with irritable bowel syndrome, often you see a co-occurrence of depression. Or it could flip it the other way around. People with depression, you often get a co-occurrence of IBS. So kind of like showing this link between brain and gut. There's also studies like this. So, for example, this is looking at 30 healthy individuals, 29 people with depression or major depressive disorder, MDD. And you see, so a lot of the times we study the microbiota by looking in people's poo, people's feces. And so we see actually people with depression have a different microbial composition than people that are healthy equivalents. And again, something we really wrestle with in science is cause or effect. Is this because the microbes are causing depression, or is it more likely that people that are depressed, lower physical activity, different diets, more stress, that's more likely to impact on the microbes? So this is something we really grapple with. So how can we untangle causality or causation? And so one of the ways this was done, this was a study coming out of Cork. So researchers in Cork do a lot of work in this field. And so just going back, something I should say about those germ-free animals we talked about. If you take an animal and deprive it of microbes from birth, then you're not just studying presence or absence of microbes, because the microbes are really important, as we talked about, for extracting calories. So even though germ-free animals eat a lot more food, they still have smaller body weights because you haven't got the microbes pulling that energy out. They also have underdeveloped immune systems. They have smaller organs. They also have lower basal metabolic rates. So it's a clean system in some ways, but it's not just presence or absence of microbes. So an alternative is this antibiotic treated model. So here what they did, they took rats, gave them antibiotics for 4 weeks to suppress their microbes, then took fecal samples from people with depression, people without depression, inoculated these rats with those fecal materials. They put those microbes into the guts, and then they did a series of tests. So they did the elevated plasmase, which we talked about earlier, and you can see those receiving the depressed microbiota had a significantly lower amount in the open arms. So they had that lower anxiety-like behavior. The open field box, looking at how much time they spend in the center, those receiving the depressed feces spent significantly lower time in the center. And this is a specific test for depression. So it's often seen with depression, something called anhedonia, which is a loss of interest in something you'd normally be interested in. And the way you can test this in rats is they like sugar, so they like sugary water. So you can put a bottle in the cage with sugary water and a bottle in the cage with just normal water and see how much they have a preference for the sugary water. So you can see those receiving the control feces spend about 80% of the time they're drinking the sugar water, significantly lower amount in those receiving the depressed microbiota. So what this demonstrates is you can actually transfer behaviors by transferring the microbiota. So it's kind of unpicking that cause or effect relationship. Now, how do we do this? How does the microbes impact on the brain? It's thought there's 3 main routes. There's this vagus nerve, which is the nerve connecting the central nervous system with the enteric nervous system, and it's known that the microbes can interact with that. There's also thought that this can happen through the immune system. So microbes can interact with the immune system at the gut, or they can move into the blood and trigger things like inflammation, which can impact on the brain through the immune system. And what we do a lot on, we study a lot, is through metabolites as well. So through the chemicals that the microbes produce that can, you're going to get shocked. So the chemicals the microbes produce, we also have been studying how these can also impact on the brain. So to go through some examples then, this is what we call the short-chain fatty acids. Again, to hammer home the point, eat a lot more fiber. So fiber gets down to the colon where your bacteria, your carbohydrate fermenting bacteria, will process that fiber and they'll make these things called short-chain fatty acids. So a lot of your Yakults, Danone type things, they usually work through this mechanism. They promote the production of these microbial metabolites, so acetate, butyrate, and propionate. And yeah, these tend to be associated with lots and lots of good things. So what we show here is that these short-chain fatty acids can bind to those receptors in the gut and trigger the release of appetite hormones. And those appetite hormones can, they increase satiety, which is the feeling of fullness. So what they do is they lower your appetite because you feel full. So that acts locally in the gut. But we also showed here in mice, so if we put a carbon 11 labeled acetate directly into the gut, we can recover about 3% of that dose in the brain. So that's a microbial metabolite produced in the gut that can actually move from the gut into the brain. And we can see once it enters the brain, it changes central processes and actually reduces appetite by acting on the brain as well. So these gut microbial signals act on the gut and in the brain to shape your appetite. And that kind of makes sense because the microbes get the majority of their food from what we eat. So it makes sense that they can control how we do it. And it's thought that this might be a mechanism to try and target obesity if we can use the microbiota to reduce appetite. Another example is what we call the blood-brain barrier, sometimes shortened to just the BBB. And that's not this kind of like a barrier around here. It actually looks more like this. So we've got capillaries right the way throughout the brain. And a bit like how we talked about the gut having those tight junctions, you've got the same thing through these capillaries. So they're nicely tightly packed, and they have a lot of control over what moves in and out of the brain. And there's data showing that the microbes can also influence this blood-brain barrier. Now here, if you consider this bit, I don't know if you can see this laser pointer or not, but on the right-hand side, this is what tends to be what a good gut looks like. You've got fiber in there, short-chain fatty acid production, maintaining these tight junction proteins, a lot of control over what moves from the gut into the circulation. Particularly as you get older, you see a more dysbiotic microbes, toxin-producing, which weaken these barriers. And that means you get this leaky gut where the bacteria or bacterial cell fragments can move from the gut into the circulation. And there's particularly types of bacteria called the gram-negative bacteria that on their cell wall they have this molecule called lipopolysaccharide. And lipopolysaccharide, or LPS, is a potent inflammagen. It causes inflammation. So if you get bacteria or their cell walls moving across, this LPS triggers inflammation. That can trigger systemic inflammation, but also neuroinflammation. So it can be a bad thing. And what we showed here, well, actually, yeah, in this study, you see one way you can measure that blood-brain barrier permeability is you can grow these cells from the blood-brain barrier, and you probably can't read that, but this is a type of dye, this fitsidextran, and you can see how much, if you put on one side, how much you can recover from the other side, and that's a measure of permeability. So you can see if you're untreated, you have this level. If you expose the cells to LPS, that level goes up. So this bacterial signal causes increased permeability in the blood-brain barrier. Another way you can test this is if you have your cells tightly packed together and you pass a current across them. If they're nice tightly packed, then you have high resistance, so you see that here, whereas if they get weaker, that resistance goes down. And so you can see when you give LPS, that resistance goes down. But if you pre-dose or bathe these cells in these short-chain fatty acids before, so particularly with butyrate and propionate, and then expose them to LPS, you don't get that rise in permeability. So these short-chain fatty acids, these microbial chemicals, can actually prevent that leaky blood-brain barrier. Another example is through neurotransmission. So you have billions of neurons in your body passing signals across. You have this cell body sends this electrical signal along the axon, and then at the synaptic button or synaptic terminal, you get the electrical signal gets converted into a chemical signal. And this is what we call the neurotransmitters. You get neurotransmitters released from here, and then they pass the signal on to the next cell. Could be another nerve, could be a muscle, could be a gland. And so these are the neurotransmitters, really important. And we know now that many of the microbes in your gut can produce these neurotransmitters. So these bacteria in your gut can actually produce a lot of these neurotransmitters. It's unlikely that the neurotransmitters produced in your gut are going to affect your brain. Is it because this is... Shall I... I'll clip it on outside, shall I? That might be easier. That's all right. I think it's because maybe it's just rubbing. If I put it there, it might be all right. Yep. So... so they're unlikely to directly affect the brain, but things, for example, serotonin. Serotonin is synthesized from the amino acid tryptophan, which you get from your diet. So if you've got a microbiome that metabolizes a lot of tryptophan, it's going to reduce the amount that's available for the host, for you. And so as a result, you're not going to be able to make enough serotonin. Serotonin is an interesting one because it's linked in with things like depression. So we think actually it's probably more like they're almost competing with you for the substrates for these neurotransmitters. I could just hold it. Oh, yeah. Start a song. Yes. Cheers. Thank you. Can you hear? Does that work? Yeah. So just to finish on, then I've got about 5 minutes. A lot of the motivation for some of the motivation for this field is in a lot of the links between autism and the gut microbiota. And this came out of the fact that a lot of individuals with autism spectrum disorders also exhibit GI symptoms. And so you can see this here. So they often have acid reflux, a compromised intestinal barrier, and also a lot of studies showing that they have a different microbial composition to neurotypical individuals. So what we think, there's a genetic susceptibility to autism spectrum disorders, but we think this also requires this environmental trigger, which can come from the microbiota. And so one of the sort of ways to demonstrate this is you can see here that the microbiota has been shown to be important for social development. So one way to test this is the three-chamber cage, where you can have these 3 chambers with a cage in the 2 end chambers. And you can put a mouse in one of the cages, and then you put your mouse in, and you see how much time they spend socializing. They're quite social animals. And you can see that in the germ-free animals, they spend significantly less time interacting with the mouse than their colonized equivalents. And then these GFCs are when you take the germ-free animal and colonize them. So you see that significantly increases the amount of time they spend with the other mouse. And then you can take this a step further. You can then put in a novel mouse and then the familiar mouse and see which one they spend more time with. And you can see in the conventional mice, they spend significantly more time with the novel mouse, but we don't see that with the germ-free animal. So you can see that the germ-free animal is impacting on that sociability. This was a study out a year or 2 ago, which also found that C-section, and we're doing some work with this now as well, because it seems that it affects the blood-brain barrier. But the C-section born mice also, compared to vaginally born mice, also have differences in their social behavior. And so this has particularly disrupted the levels of bifidobacteria in the gut of these mice, and actually shows that you can rescue that behavior, that lack of sociability, by co-housing them with other animals, so you get this transfer of microbes, or giving things like probiotics or prebiotics, which I'll discuss in a minute. And so we think this might be mediated by, we've shown that the microbiota of individuals with ASDs or autism spectrum disorders tend to produce less GABA, which is a neurotransmitter. And we also think this is acting via a blood-brain barrier mechanism. So hopefully I've convinced you there that the microbiome is super important and also can impact on your brain. So the next thing is, can you change it? And this really is why it's a really interesting area, because the accessibility of the microbiome in your gut means you can actually change it through relatively easy, simple, manageable approaches. And so these can tend to be through nutrition-based approaches. So we have prebiotics. So prebiotics are kind of similar to the mechanism to the fibers we talked about. They are usually sugars that can make it past your gut and feed. They stimulate the growth of beneficial bacteria that are already in your gut, in your colon. We have probiotics, which are what you might see in the supermarkets. These are like usually yogurt-based solutions that have really high levels of beneficial bacteria. And they need to be really high because when you take them in, they need to make it past your stomach, past your small intestine, back into the colon where it'll have its effect. Now, these aren't usually found in your gut, so they're quite poor competitors. And so as a result, you flush them out quite quickly, which is why you have to take them every day. Whereas prebiotics, you're stimulating what's already in there. Symbiotics are a combination of the two, and there's a new brand or new class of these as well now called the postbiotics, which is where you can take probiotics and heat treat them, is one example, to kill them. but they still have the beneficial effects. So, and we think that's through either their metabolites, cell wall fragments, or certain compounds. So that's postbiotics, so it's like the dead probiotics. And we've shown, I'll skim over this one, but we've essentially shown in rats, giving these prebiotics, that we can actually reduce anxiety-like behavior. I'm just really hammering home the fiber point here. You should have 30 grams, but most people consume 20. It's probably not even that much. Take more fiber. Finished then. This is the sort of new wave in therapeutics for your microbiome, the fecal microbial transplants, or FMTs as they're often shortened to. And so the C. diff example I introduced earlier, that's probably where this is most effective. This idea that if you don't have an established sort of healthy microbiome, C. diff can get in, and it's very hard to get rid of. And if you've had diarrhea for 6 months, then this suddenly becomes like quite an attractive option. The idea here is you take the feces from a donor and you transplant that into your gut. And so that restores a healthy type microbiota. And so how it works, so you start by taking antibiotics. You then take polyethylene glycol, which is a laxative, helps flush out the system. And then you take feces. So you get to pick your donor. So just something to think about tonight. Usually it's a spouse because you share the same diets. And you can go from the top or from the bottom. Yeah. So, and that fecal slurry goes in, and actually the results are very, very effective, particularly for C. diff. So this is, sort of really pushing the boundaries, sort of therapy for this area. And in fact, we don't fully know exactly the mechanisms behind it, because there's some data showing that even if you heat treat, so kill off the bacteria, it's still effective, so it could be viruses. There are companies now trying to make synthetic FMTs, so actually trying to build sort of synthetic communities of like 10 to 12 bacteria, and also showing pretty significant effects. So this is definitely an area for the future. I think there's even some clinical trials looking at this in depression as well. And then, so they were how you can promote beneficial bacteria. There's also some processes now to try and inhibit harmful bacteria. So, for example, this was looking at adolescents with autism spectrum disorders. This research group found that these particular microbial metabolites could increase anxiety and irritability in people with ASD. So they developed these chemical binders, a bit like charcoal. So when you take them orally, they selectively bind to these microbial metabolites in the gut, stopping them from being absorbed. You then excrete them in the feces, and that reduced the amount of these metabolites in the blood. And what that did, it actually reduced anxiety and irritability in these individuals. We've also been playing around with these chemical inhibitors that can actually bind to microbial enzymes and stop those enzymes having their function. So in this particular study, we found that microbial activity on a particular chemotherapeutic increased its toxicity, and showed that if you block that enzyme, you reduce that toxicity, so you can actually increase the dose of this chemotherapy dose. So there's lots and lots of growing areas to try and manipulate this, and I think I will end there, a few minutes over, sorry about that. But yeah, happy to take questions, thank you.